Hello everyone, I'm Alexandre Boulay. I'm a master's student at Laval University in Quebec City, and my project is on predicting the bacterial hosts of bacteriophages using artificial intelligence. So first of all, what is a bacteriophage? It is a virus that specifically infects bacteria. It possesses mechanisms that allow it to recognize, bind to, overtake, and eventually kill its host. Why is it important to study them? To better understand their interactions with bacteria, their impacts on our health, and most urgently, to use them to develop treatments against multi-resistant bacteria. So the main objective of my project, as seen in point 2, is to build a tool to predict the bacterial host of a phage using the proteins the phage possesses to make our predictions. In other words, we are trying to predict which phage infects which bacterium. We realized along the way that it would also be useful to have the functions of these proteins at our disposal, so we also built a tool for that task. To create our dataset, we downloaded phage genomes with known bacterial hosts and clustered them. Phage proteins were predicted using PROCA and annotated using a model we trained ourselves. Finally, protein embeddings were obtained using PROT-T5. These are representations of the proteins that we can feed to machine learning algorithms. The training label is the host of the phage that possesses this protein. Here is the hierarchical model we built. Inputted in our model are all the proteins a phage possesses. In this example, I chose to input a subset consisting of all phage lysins. They are proteins that degrade the bacterial cell wall. Using these proteins, we first predict which type of bacteria the phage infects, so either a gram-positive or gram-negative bacterium. Then, we use a second model to predict the bacterial host of the phage at the genus level. Obviously, a phage possesses more than one protein. To take this into account, one prediction is made per protein, and the probabilities associated with each host are averaged. And the most probable host is assigned as the final prediction. In reality, keep in mind that we have way more than 2 hosts, which complicates the training of our models. An extra challenge arises as we try to create a test dataset, as phages share many proteins, like we can see in the graph. This may lead to an overestimation of performances if we are not careful. More naive ways of separating the data will suffer from this problem. However, at the opposite end of the spectrum, clustering algorithms that place very similar phages in the same test or train set may not be representative of the diversity of the phage population. A compromise must be made, or perhaps both methods must be compared to obtain a better picture. Let's now look at the results. To begin with, the first layer of the model works very well. We are able to predict if a phage infects a gram-positive or gram-negative bacterium nearly perfectly. But this isn't a surprise, as the task itself isn't that complicated. As we can see in the figures, the proteins from gram-positive and gram-negative infecting phages differ a lot already in 2 dimensions. Next, let's look at the bacterial host predictors. These classifiers are more precise as we are attempting to predict the genus of the host. For example, Klebsiella, Vibrio, or Salmonella. Here, I am comparing the results for 4 models. Models trained either using structural lysins or endolysins produced inside the bacteria for both gram-positive and gram-negative infecting phages. We can first see that lysins are better predictors for gram-positive infecting phages. This is likely because the sugar coating around gram-positive bacteria is more diverse and more complex, making the phage lysins more specific and hence more useful as predictors. In other words, a greater diversity between genera results in better predictors. Second, we notice that virion-associated lysins are better predictors for gram-negative infecting phages than endolysins. Again, we suspect this has to do with the fact that the most external layer of sugars is more diverse than the inner protected layer. Next, we wanted to see if there's a way to improve these results, especially for gram-negative infecting phages. We thus trained binary models for each host. The example I'm showing here is a model trained to predict Gordonia phages, for which we managed to obtain performances of 99%. The blue bar shows the confidence of proteins that led to correct predictions, and the red bar shows those of incorrect predictions. And I separated these by protein function. The most important result here is that base plates and tail appendages are the most discriminatory and should be the focus, at least I believe, for the next models we train. We can also see that Holins, integration-related proteins, and annealing proteins are also promising. We also notice that endolysins are not as good predictors for Gordonia, which is surprising as it is a gram-positive bacterium. To recap, we trained multi-class models using lysins that work pretty well for gram-positive infecting phages. However, these models can be significantly improved, especially for gram-negative infecting phages, by training binary models. This allows us to adapt our models to the bacterial host in question, depending on if it is a gram-positive or gram-negative bacterium, or if it has distinctive proteins, for example. The objective, of course, is to be able to obtain a tool we can use to study phages, to better understand the microbial interactions between them and bacteria, as well as the impacts they have on our gut health. Taking things further, models like these are the first step to using phages to develop treatments against multi-resistant bacteria. Thank you for listening, and looking forward to meeting you in person. A big thank you to the members of my lab, Professor Rousseau, as well as all of my colleagues.